Welcome to this explainer. Today, we're embarking on this incredible journey straight into the human body. We're going to travel from the massive, macro-level engineering of your heart all the way down to the microscopic physics of the blood itself. It's just the stunning interplay of plumbing, physics, and fluid dynamics that literally keeps us alive every single second.
Here's our roadmap for today. We'll start with the evolutionary plumbing problem, then examine the physics of blood flow. After that, we'll cover measuring the hidden system, and finally, deconstruct the fluid itself by asking, what exactly is blood?
Okay, part 1, the evolutionary plumbing problem. To truly appreciate our cardiovascular system, we first need to understand the fundamental biological challenge of physical size.
So how do nutrients actually reach cells buried deep inside a large organism? Well, if you're an isolated single cell or just a really tiny organism, you don't even have this problem. You can rely on simple diffusion to pull nutrients in from your environment and push waste right back out. But humans, we are way too big for that. The distances between our external environment, like the air in our lungs or the food in our gut, and the cells deep inside our core, well, they're just too vast. If we relied on a simple open-ended tube, all those nutrients would be completely depleted by the time they reach the deepest cells. So nature came up with a brilliant evolutionary solution, an integrated, high-speed circulatory system that uses convection to rapidly move fluid around. And the engine driving this whole solution, that's your heart, which is actually a dual pump. The right side boosts blood to the lungs, and the left side drives it through your entire systemic circulation. Get this. Over an average 75-year lifetime, your two ventricles combine to pump a staggering 400 million liters of blood. To put that into perspective, that is enough fluid to fill an entire lake. We're talking a lake a full kilometer long, 40 meters wide, and 10 meters deep. It's an absolutely massive, continuous feat of biological engineering, constantly fighting against gravity and physical resistance. Which brings us to part 2, the physics of blood flow. Now that we have a massive pump pushing fluid, let's look at how that fluid actually behaves inside your vessels. To really understand blood flow, we look to Poiseuille's law. This principle states that fluid flow through a rigid tube is directly proportional to the fourth power of the vessel's radius and inversely proportional to its viscosity. The absolute crucial point here is that fourth power part. Flow is incredibly sensitive to the size of the vessel. If a blood vessel's radius increases just a tiny bit, the flow increases exponentially. Conversely, even a minor narrowing massively restricts the flow of blood. This is exactly why even a tiny amount of plaque buildup in your arteries makes your heart work exponentially harder. You see, resistance to flow comes primarily from the microscopic geometry of the vessel, not just the properties of the blood itself. And this brilliantly illustrates how blood actually travels within those vessels. You basically have two potential types of flow, laminar and turbulent. Laminar flow is the ideal state. It's smooth, it's silent, and it's highly energy efficient. The blood moves in these organized, concentric layers, with the center layer moving the fastest. But when things go wrong, or the velocity spikes, you get turbulent flow. It's chaotic, it wastes kinetic energy, and crucially, it is noisy. That noisy turbulence is exactly what doctors are listening for when they detect heart murmurs or arterial bruits. The chaotic vortexes of blood literally vibrate your surrounding tissues, creating a sound you can actually hear with a stethoscope. So you might be wondering, how do we know when that healthy, silent flow will break down into chaotic noise? Well, we actually have a mathematical way to predict that, called the Reynolds number. This is a dimensionless quantity that factors in vessel radius, blood velocity, and viscosity. When the Reynolds number is below about 2,000, flow remains smooth and laminar, but as it pushes past 3,000, turbulence totally takes over. This number spikes when vessels get abnormally large, like in the aorta, or when blood velocity shoots up, maybe due to a narrowed heart valve. If you've ever wondered how doctors know the physical limits of your circulation, it comes right down to this exact equation. Now, let's consider the viscosity of the fluid itself. Water flows linearly under force. Honey also flows linearly, it just has higher viscosity. But blood is a non-Newtonian fluid. It actually behaves a lot more like mayonnaise. Yes, your blood is basically anatomical mayo. Just like mayonnaise, blood requires a specific amount of force, what we call a yield shear stress, just to start moving. If you gently tip a jar of mayo, it just stays put, right? Blood does the exact same thing at very low forces. This happens because of these really complex structural interactions between red blood cells and a plasma protein called fibrinogen. These proteins cause the red blood cells to stick together slightly, creating a resistance that must be overcome before flow can even begin. Moving on to part 3, measuring the hidden system. We know the physics now, but how do doctors actually measure these extreme internal forces in a clinical setting? You have very likely experienced this process yourself with a blood pressure cuff, formerly known as a sphygmomanometer. When a clinician wraps that cuff around your arm, they are literally utilizing the exact physics of turbulence we just talked about. First, they inflate the cuff above your highest pressure, your systolic pressure, to completely pinch off the brachial artery. Next, as they slowly release pressure, it drops just below your systolic peak, and a spurt of blood escapes under the cuff. This rapid movement creates turbulence, which makes a sharp tapping noise known as a Korotkoff sound. Finally, they keep releasing pressure until the sounds muffle and completely disappear. That silence, that means the artery is fully open again, and laminar silent flow has returned, marking your lowest or diastolic pressure. For a more advanced look at these internal dynamics, cardiologists use a Doppler echocardiogram. Using the exact same principle police use in radar guns to catch speeding cars, the ultrasound bounces off your moving red blood cells. By analyzing the change in frequency, the machine color codes your blood velocity. Red indicates flow moving toward the ultrasound transducer, and blue indicates flow moving away from it. It gives doctors this incredible, real-time, highly detailed map of the exact direction and speed of flow through your heart valves, and they can do it all without making a single incision. And that brings us to our final section, Part 4, What exactly is blood? We've talked about the pump and the pipes. Now let's deconstruct the fluid itself. If you take a whole blood sample and spin it in a centrifuge at high speeds, it separates out into 3 distinct layers. The top fraction is the plasma, which is a pale, watery solution of electrolytes and proteins, making up about 55% of the total volume. The heavy red blood cells sink to the bottom, forming the hemocrit layer, which is around 40 to 45% of the volume. But the absolutely crucial point here is what lies sandwiched right between them. It's a tiny, whitish layer making up less than 1% of the volume called the buffy coat. Despite its tiny size, this layer is incredibly important because it contains almost all of your white blood cells and platelets. Let's look a little closer at the proteins floating in that plasma using a technique called electrophoresis. Notice that huge, dark band on the left side of the plasma sample? That's fibrinogen. It's the massive protein responsible for blood clotting, and yep, it's the exact same protein we mentioned earlier that gives blood its non-Newtonian, mayonnaise-like yield shear stress. Now look at the serum sample on the right. That fibrinogen band is completely missing. Why? Well, because serum is simply the fluid left over after a blood clot has already formed. All the fibrinogen has been used up to build the structural strands of the clot itself. So where does all this complex cellular material even come from? It's a continuous process called hematopoiesis. All of the cells in your blood are born right in your bone marrow from multipotent stem cells, and they're guided by chemical signals called cytokines. For instance, if you travel to a high altitude, your kidneys release a hormone called EPO, specifically triggering the creation of more red blood cells to meet your body's increased oxygen needs. Other signals stimulate giant cells called megakaryocytes to literally butt off tiny, nucleus-free fragments. Those fragments are your platelets, constantly patrolling your bloodstream, just waiting to trigger a clot if a vessel breaks. The red blood cell itself is an absolute masterpiece of biological engineering. To maximize its ability to carry oxygen, an emerging red blood cell actually ejects its own nucleus and mitochondria. It trades off long-term survival for storage capacity, packing itself with an incredibly dense concentration of hemoglobin. And because it has no mitochondria, it survives purely on basic glycolysis for energy. Furthermore, it adopts this really unique biconcave disc shape. This geometry drastically increases its surface-to-volume ratio, perfectly optimizing the rapid exchange of oxygen and carbon dioxide gases as it squeezes through your tiny capillaries. Meanwhile, the white blood cells, or leukocytes, make up your dynamic internal defense system. Neutrophils are your first responders, actively hunting and eating bacteria. Eosinophils deploy toxic proteins against parasites and help manage allergies. Basophils act as the alarm system, releasing histamine. And your lymphocytes? They are your advanced, targeted immune memory, with B cells acting as highly specific factories for antibodies. Finally, monocytes patrol the blood before migrating into tissues to mature into large, debris-eating macrophages. It is literally an entire, highly coordinated army flowing right through your veins. Which brings us to our final thought. Knowing the incredible physics, biology, and chemistry at play, can you feel the turbulent, microscopic universe rushing through your veins right now? Think about the fact that every single second, this staggering interplay of hydrostatic pressure, non-Newtonian fluid flow, and specialized cellular defense is working relentlessly to keep you alive. It is a true miracle of evolutionary engineering. It really makes you wonder, what other hidden physiological wonders are waiting to be explored inside you? Thanks for joining me on this explainer, and keep learning.